Okay, so welcome to this next video in the playlist on lipid modifications of proteins. Okay, right. So in this video what we want to do is discuss the detection of palmitoylation and the method for which we are going to use to detect protein palmitoylation is tritiumated palmitic acid. Okay, so detection of protein palmitoylation with tritiumated palmitic acid. Okay, so the structure for this video then, we're going to start with a discussion of tritium basically, and its radioactive properties. We're then going to move on to the methods by which you can actually use tritiumated palmitic acid to detect the palmitoylation of your favourite palmitoylated protein basically. Okay, so right, let's start by discussing tritium then. Okay, so tritium is an isotope of hydrogen. Okay, so let's start off with what hydrogen is. Okay, so hydrogen atoms are atoms which have one proton at the center basically. So in their nucleus they have one proton, and therefore they also have one electron orbiting them. Okay, so that is the that is what you have to be to be called hydrogen basically. You have to have a proton, and I'm going to colour this in a certain colour. So I'll colour this in vivid purple here, and this represents our proton. Okay, so hydrogen atoms have one proton and one electron. The question then becomes, how many neutrons do they have? Well, basically, the number of neutrons is not set. Okay, so in essence, you can have any number of neutrons you like, and it would still be named hydrogen, okay? But obviously the different hydrogen atoms, which have different numbers of neutrons in their nucleus, will have different properties, okay? So these are called the different isotopes of hydrogen. So they all have one proton and one electron, but they have differing numbers of neutrons. Okay, so the most common isotope, isotope that should be. Okay, so the most common isotope of hydrogen is protium. Now, protium, basically you have no neutrons. So if you have no neutrons and you just have one proton and one electron, that's a protium atom, okay? Most common form of hydrogen is protium, especially on the planet. Basically, people will usually just say hydrogen. They won't specify that they're dealing with protium. So if we're dealing with a protium atom, generally people will just call that hydrogen. So if someone says hydrogen and doesn't specify which isotope they mean, you can assume they mean protium. Okay, then the next isotope up is deuterium. Okay, so deuterium is another isotope of hydrogen. And deuterium atoms have one neutron at their center basically. So they have one proton, one electron, and one neutron. Okay, and then finally, up from deuterium, you then have tritium. Okay, and this is the one that's of interest to us. And tritium atoms have two neutrons in their nucleus. So they have one proton, two neutrons. So I'll draw this in now. Okay, so here are the two neutrons, which I'll colour in blue, and then one electron also orbiting it. Okay, now neutrons are neutral basically, they don't have a charge, whereas protons are positive, so the tritium atom is still neutral basically. Now, let's discuss the decay of tritium. What does it decay into? Basically, tritium is a radioactive atom, okay? So it's not stable, it's going to undergo a radioactive reaction and release radioactive molecules, basically. Okay, so the type of reaction that tritium is going to undergo is one of these neutrons is going to turn into a proton. Okay, so what's going to happen is the atom is going to go from being a single proton with two neutrons and one electron to being two protons and one neutron. with an electron surrounding it. Okay, right. So now here's one neutron, and here's the 2 protons that we now have. Now this is a different atom. Okay, and you might say, well, don't you need another electron? Well, atoms all the time have electrons removed from their outer shells, and it doesn't change what element it is. So fundamentally, what decides which element you are is how many protons you have. So this atom now has 2 protons, okay? And the atom with 2 protons, however many neutrons you have, the atom with 2 protons is helium, basically. Okay, now, people will generally write, and I should have discussed this when I was discussing tritium, people will generally write tritium like so. They'll say H3, and if they're being really precise, they'll put a 1 there as well. So these 2 numbers here, the 3 here is the mass number. Okay, so this tells you how many particles you have in the nucleus. So this tells us that we have 3 particles in the nucleus. This number down here is the atomic number. Okay, and this tells you how many protons you have. So hydrogen just has 1 proton. So basically, if we were doing the same thing for helium, what we'd do is we'd have the symbol for helium, and our mass number is 3, and then our atomic number now is 2. We have 2 protons, which is why we've changed element from hydrogen to helium. Okay, so often people omit the atomic number because the name of the element tells you how many protons you've got, so it's a bit of a redundant information there. But you should put the mass number up there, because that tells you which isotope of this element that you're dealing with. Okay, so people will often just denote tritium as H3, superscripted. Right, okay, so what's going to happen is a neutron within the tritium atom is going to turn from a neutron into a proton. Now, when you do that, you've gone from being a neutron, which was neutral, to being a proton, which is positively charged. So you're also going to have to release a negatively charged particle. So what also happens is you release an electron. And I'll just show this as a cross, since I showed these electrons here as crosses. Okay, so you release an electron, and often the symbol for an electron is E with a negative sign up there. It's also known as beta negative radiation, okay, or a negative beta particle. Okay, but effectively this is just an electron being emitted. Now you also emit something else which we're not going to dwell on hugely, but I should just state it to be correct. You also emit something called an electron antineutrino. Okay, so this sort of V symbol means neutrino, the bar means antineutrino, and the E there means that it's an electron type antineutrino. Okay, so this is an electron antineutrino. So you emit 2 particles, you emit an electron, and you also emit this electron antineutrino over here. Now, as I say, we're not going to dwell on this at all. I've just put it there to be complete. We're going to look at this because this is basically extremely inert. It can, you know, you've got these things passing through you all the time and they do nothing. So this isn't what you're going to detect, basically. It's the electron that's being emitted that you'll detect. Okay, so this is what's known as beta decay, basically, or beta radiation. So when you emit electrons, that's called, often people will just call it beta radiation. If you're being very precise, you'll call it beta negative radiation, because there is also beta positive radiation where you emit a positron rather than an electron. Okay, so we're going to detect these electrons that are coming off the tritium atoms. And of course, if you've got a huge number of tritium atoms, not all of them will be decaying at once, but a handful of them will be decaying. So it's effectively spontaneous when a tritium atom decides to decay into a helium atom. So if we write out now the entire formula for it, we start off with hydrogen, and I'll put the mass number as well as the atomic number, okay, and it will decay down into helium like so. So that's what's happening there, is that one neutron is turning into a proton, okay, and it will also emit an electron plus this silly thing called an electron antineutrino. Right, okay, so that's the decay of tritium. Now, how are we going to use this to detect palmitoylation of proteins? Well, basically, you can detect tritium by just looking for the emission of electrons. You can detect this with, for instance, a Geiger counter. Okay, so what we're going to do is create a radioactively labelled form of palmitic acid. And the form we're going to create is called 910. And then you put tritiumated, so you put a tritium atom at the front, so you put H3 for tritium, okay, and then you put palmitic acid. Now I should just say that tritium is often just abbreviated to T for short, but we'll also use H3 rather than T. Okay, so let's draw the structure of 910H3 palmitic acid. Okay, so palmitic acid is a 16-carbon fully saturated carboxylic acid. Okay, so here is the carboxylic acid group, and now it's going to require a bit more thought with drawing out the structure because we've got special things on 9 and 10. Okay, so we then need 15 more carbon atoms. Now the first 7 are just going to be attached to normal hydrogen atoms, and these will just be protium atoms, okay? So they'll just be a proton with an electron rather than tritium atoms. So here are the first 7 of these methylene groups. Now, because I don't want to have to draw out 7 methylene groups that are identical, what I've done is I've drawn one, put a bracket, well, put 2 brackets around it, and then put a subscript 7 here. Okay, right, so that now takes us up to the 8th carbon, because here's the first, then we've got 7 more, so we have got to the 8th. The next one will be the 9th, okay, so we have to draw this, because this has got a special hydrogen atom coming off it. So I'll put H3 here, to show that it's got a tritium atom coming off it, and then it will have 1 protium atom coming off it. Okay, the 10th carbon also has a tritium atom coming off it, and then 1 protium atom coming off it. Okay, now it's simple for the rest of it. We just need to complete up now. So we've done 9 so far, 10. We've got 6 more to go. So 5 more methylene groups, which are normal methylene groups, where you've got protium off these carbons. Okay, so here's our 5 other normal methylene groups, and then right on the end we've got a methyl group. And I'll have to show that separately. Okay, so here now is the structure of 910 tritiumated palmitic acid, and for short, you would just say, the way that you would pronounce this, the way that you would read this rather, you would read it as tritiumated. Okay, so you would call this 910 tritiumated palmitic acid, or for short, you might just call this tritiumated palmitic acid. Okay, right, so now let's see how we're going to actually do an experiment with 910 tritiumated palmitic acid to determine whether a certain protein is palmitoylated. Okay, so let's say we have some protein, okay, which I'll just call protein A to keep it completely arbitrary, but this is not meant to in any way suggest that this is protein kinase A. It's just an arbitrary protein that I've called protein A. Okay, and let's suppose we want to find out whether protein A is palmitoylated. Okay, so let's imagine that it is going to be palmitoylated, and then let's think through what the outcome of the experiment we're about to do will be, and then we'll think about what would have happened if it wasn't palmitoylated, and we'll see how the result would have been quite different, and therefore how this experiment allows you to determine whether protein A is palmitoylated or not. Okay, right, so the first thing to say is that you are going to start with a cell line of some sort, and you are going to transfect in an extra gene for protein A, and the reason you're going to do this is that you want a huge overexpression of your protein so that the experimental results are very clear, basically. So you've got a large amount of the protein to be working with. even though the cells will probably have a gene, in fact they'll probably have 2 genes for protein A already, we want protein A hugely overexpressed, and the best way of doing this is to add in a new gene, an extra copy of the gene, okay, by putting it on a plasmid, for instance, and transfecting the plasmid into your cell line. Okay, so I'm going to start off with a discussion of some decent cell lines that you could use to do this experiment in. So, certain cell lines that are common cell lines to be used. Firstly, we'll start off with 2 fibroblast-like cell lines. The first is called COS1, okay, so COS1 cells. Okay, the next is called NIH, okay, and then 3T3 cells, so NIH 3T3 cells, and sometimes NIH 3T3 cells are just called 3T3 cells for short. Okay, now both of these cell types are very similar to fibroblasts. Remember, fibroblasts are these very long, thin cells which produce connective tissue, okay? So if you were at any sort of connective tissue portion of the body, for instance maybe a tendon, you'd find a lot of fibroblasts there, or a scar tissue, you'd find a lot of fibroblasts. Okay, so here's its nucleus. So these cells are fibroblast-like in nature, and they're common cells to be used in experiments. So they're fibroblast-like. Okay, so we'll look at the names of 2 other cell lines, which this time are epithelial cell-like. Okay, so a very famous cell line now, Mdc K cells. Okay, and what does MDCK stand for? Well, basically it's a bit of a joke name. Okay, so it stands for, M stands for maiden, okay, D stands for Derby, so maiden Derby, and then CK stands for canine kidney cells. Okay, so this cell line was originally extracted from Cocker Spaniel dogs, basically. It's a canine kidney cell, and of course many of the cells of the kidney are epithelial-like in nature. Okay, so here's a little epithelial cell to remind you. Okay, now there's another important epithelial cell-like cell line, which is known as 293 T cells. Okay, so 293 T cells, and these basically are a modified, better version of HEK 293 cells, and HEK stands for human embryonic kidney cells. So again, it's extracted from the kidneys originally of a human embryo. Okay, but of course you don't have to extract them from human embryos forevermore. Originally it was from human embryos, but now we've just propagated the cell line on and on so that you don't have to continuously use human embryos, because there are ethical and cost issues with that. Okay, so human embryonic, it should be embryonic kidney cells. Okay, so the 293 T cells are a modification of these HEK 293 cells, and they're all, 293 T cells as well as maiden Derby canine kidney cells are both epithelial cell-like. Okay, so they're similar to epithelial cells. Right, so you can pick whichever cell line you like, and what we're going to do is transfect these cells with an extra copy of the gene for protein A, so that the cell hugely overexpresses protein A and is therefore perfect for us to do experiments with. And now what we're going to do is get the cell to add on palmitoyl groups, which are radioactively labeled. Basically, we're going to get them to add on tritiumated palmitic acid. Okay, and we'll continue this discussion in the next video.